Each of us shares the characteristics of our species, but we also have individual differences. We are male or female. Some of us have red hair, others brown eyes. Some are tall, others short. For a long time, it's been known that these characteristics are passed from one generation to another. But how is it possible for hereditary information to be copied in such a way that characteristics of the species are passed from one generation to the next, and yet individual differences are expressed? Questions about heredity have led to an unraveling of what some have called the blueprint of life, perhaps the greatest accomplishment of biological science in the last 50 years. Scientists now know that multicellular organisms like plants and animals, such as humans, contained in the nucleus of each of its cells in a molecule called DNA, deoxyribonucleic acid. The DNA molecule is made up of two long strands of sugar and phosphates that provide a type of backbone for the molecule. The two strands are twisted into the shape of a double helix or spiral. The two strands are bound together by pairs of bases resembling a ladder. The molecule is so long it is coiled and folded many times. Each DNA molecule is packaged into structures called chromosomes within the nucleus of the cell. Chromosomes vary in size and shape and usually occur in matched pairs. The number of pairs of chromosomes in a cell depends on the species. Goldfish, for example, have 47 pairs. Orchids have 28. Guinea pigs have 32. And shrimp have 127 pairs. Humans have 23 pairs for a total of 46 chromosomes in each cell. In many species, including humans, there is a pair of chromosomes that determine the sex of the individual. Females have 2 X chromosomes, while males have an X and a shorter Y chromosome with fewer genes. Each chromosome is composed of a well-defined mixture of protein and DNA. The protein is a special type that makes spools, and the DNA wraps twice around each spool in a series of double loops. If the DNA molecule could be unraveled, it would show 2 long strands, such as this. Each strand of DNA consists of a series of units called nucleotides. These nucleotides make up the genes that contain the hereditary information of the individual. The nucleotides are made up of chemical compounds utilizing only 4 bases: cytosine, represented by the letter C, thymine, T, adenine, A, and guanine, G. These bases only occur in certain combinations. A only goes with T, and C only goes with G. Long sequences of hundreds or thousands of these bases make up one gene. Genes control everything because they comprise the codes for all the proteins that orchestrate and regulate every operation of the cell. These proteins come in a wide range of shapes and sizes and have many functions. Some are enzymes that catalyze chemical reactions. Other proteins are involved in the digestion of food. Others carry oxygen in the blood, and still others build cells and tissues. Proteins are absolutely essential for life. 4 letters or chemical compounds may not seem like a big alphabet to make all of the amino acids and proteins necessary for an organism. And yet the combinations and the way the combinations are sequenced are more than enough. Just these 4 compounds make up the codes for 31,000 genes, all the information in the human genome, and yet they provide the uniqueness of each individual. Except for identical twins, Every individual has DNA that is slightly different from every other individual in the species. It is those differences that give us our individual characteristics. When cells divide, it is vital that all the genetic information is passed on to the new cells. Most cells in plants and animals use a process of cell division called mitosis. During mitosis, one cell divides to become two cells. There are 5 stages in the mitosis process. In the interphase, the DNA double helix unravels and comes apart like a zipper. Each strand then replicates itself exactly so that the nucleotides and gene sequences are repeated exactly in the new strand. This results in 2 copies of each chromosome called sister chromatids. The cell now contains 2 complete copies of its genome. During the prophase, the sister chromatids condense and thicken. At this stage, the chromosomes are now visible, as shown in this photograph taken by a light microscope. The sister chromatids line up along the central axis of the cell during the metaphase. At the beginning of the anaphase, the sister chromatids separate at the centromere, and each half of the pair then moves towards opposite poles of the cell. In telophase, the final stage in mitosis, a nuclear membrane forms around the chromosomes at each pole, and the cell divides at its equator. 2 new cells have been created, each with a matching full set of chromosomes as well as an identical complement of cellular structures. Single-celled prokaryotic cells, such as bacteria, only reproduce themselves using mitosis. In this process, 2 new cells are created that are genetically identical in every way. If multicellular organisms only used mitosis to reproduce, all of their offspring would be exactly the same, because their genetic information would be identical. Consequently, there would be no variety in the species. Multicelled organisms, such as plants and animals, reproduce sexually. 2 sex cells, one from the male and one from the female, are fused to make one new cell with half of the genetic material coming from the father and half from the mother. This is called fertilization. With meiosis, a special type of cell division occurs in the reproductive organs of both the male and the female. As in mitosis, the chromosomes replicate themselves, thus doubling the genetic material. Each chromosome now resembles an X shape, formed by 2 sister chromatids. But then something different happens. Each chromosome lines up with its corresponding partner and they exchange genetic material. This is known as crossing over. Now the chromosomes collect in the middle of the cell, and 1 chromosome from each pair is pulled to opposite ends of the cell. The cell divides in 2, both containing half the number of the still attached chromatids. Each of these cells now divides, splitting the chromatids in half. By the end, we have 4 gamete cells, each containing half of the necessary DNA. If these cells were produced by a female organism, the gametes will be the ova or egg. If by a male, the sperm. When fertilization occurs, the egg fuses with the sperm and the 1/2 of the DNA from the mother merges with the 1/2 from the father, making a complete set of chromosomes. Once union has occurred, this single cell then divides through the process of mitosis and grows into an adult. Human beings, for example, begin as 1 cell and grow to become adults of approximately 100 trillion cells. The incredible number of gene combinations that results from this type of union ensures the genetic diversity of the species and the uniqueness of every individual. For centuries, it has been understood that individuals inherited traits from both of their parents. But the evidence was so complicated and contradictory that it was difficult to determine the patterns of inheritance. In the 1850s, Gregor Mendel, an Austrian monk, conducted a series of experiments on garden peas that led to the foundation of modern genetics. Mendel systematically crossbred pea plants with different characteristics. Some had smooth peas and some wrinkled peas. Each time he crossbred plants with smooth peas with those that had wrinkled peas, he found that all of the resulting plants produced smooth peas rather than a blend of the traits. But the next generation produced some wrinkled and some smooth. Something was carrying these factors on to following generations. By careful study, Mendel came to some very interesting conclusions. When an organism has two different traits, one of the traits is dominant and the other recessive. In the case of pea plants, the trait of round peas is dominant and the trait of wrinkled peas is recessive. Mendel also discovered that while the dominant trait might be expressed in one generation, the recessive trait could be expressed in a later generation. As another example, in humans, brown eyes are dominant over blue eyes. So how can brown-eyed parents have a blue-eyed child? The answer is that both parents may carry the dominant gene for brown eyes and a recessive gene for blue eyes. If either passes on the dominant gene, the child will have brown eyes. But if both parents pass on the recessive gene for blue eyes, the child will have blue eyes. We now know that Mendel's inherited factors are the result of the absence or presence of these traits in genes. Each parent contributes half of the genetic material to the new organism. The thousands of combinations of genes and dominant and recessive traits that are possible make each individual unique. In all the copying of the thousands and thousands of base pairs of nucleotides during cell division, it is possible for errors to occur. When these types of mutations occur, serious problems can result. Cystic fibrosis occurs when a certain part of the gene is incorrect and the protein misfolds. Down syndrome is the result of an extra copy of chromosome 21, resulting in a total of 47 chromosomes rather than 46. Sickle cell anemia is a genetic disorder brought about by a one-letter misspelling of a base pair in a gene that results in defective red blood cells. Cells have ways of correcting errors. A type of proofreading function scans the DNA and corrects any base pairing mismatches by removing the improper nucleotide and replacing it with a proper one. Some mutations occur spontaneously, but others can occur as a result of exposure to physical or chemical agents. Cancer is a mutation of the cell mechanism that regulates cell growth. It can be caused by such things as cigarette smoke, ultraviolet rays from the sun, exposure to asbestos, medical X-rays, and other natural and environmental causes. Once a cell has become cancerous, it can divide, repeating the cancer in each new cell that is created, which spreads the disease. But not all mutations are destructive. Over the course of evolutionary time, mutations in the cells of organisms have led to genetic diversity and the rise of new and more competitive species, better adapted to their environment. If every cell in an organism has exactly the same genetic information as every other, how is it possible to develop the great diversity of different types of cells that are in the body of any multicellular individual? Plants have leaf, root, and trunk cells that are structurally quite different from each other. The human body has approximately 260 cell types. This is only possible through sophisticated gene regulation of the cells that help individual cells within an organism develop and function in a specialized way. Shortly after conception, the embryo has many stem cells that have the ability to develop into different kinds of cells. As the organism grows, cells become specialized as nerves, muscles, skin, or blood. Others become organs with specific functions, such as the kidneys, heart, eyes, or lungs. It is the genes that control this process. All of this complicated differentiation of cells is built into the genetic code of every cell of the organism. In 1990, scientists from around the world began the most ambitious research effort in the history of biology, the Human Genome Project. The program's goal was to identify and map the sequence of all DNA in human chromosomes. The reason the project was undertaken was to understand the regulation of genes in the cells and how this leads to the complexity of cell structures and functions. Scientists hope to use this information to develop treatments for a variety of genetic disorders. In June 2000, after just 10 years of work, 2 teams of researchers announced that they had completed mapping the human genome. Both published their findings in February 2001. It had been expected that there would be over 100,000 human genes, but both teams found only 31,000. Scientists now have a detailed blueprint of our genetic code, which will enable them to look for patterns and relationships among the different genes. This research on cells is forcing us to look at serious ethical issues. It is now possible to genetically modify plants, animals, and even humans. Genetically modified foods have been recently developed, and there is concern that these new plants, with resistance to disease, may overwhelm native plants. Cloning of human beings is now a possibility, and it may be possible to select specific traits in our children. What are the long-term implications of these developments? Should individuals be allowed these choices? All of us will have to face these ethical issues in the future, and our decisions could have long-term implications for the environment, the human race, and life on the planet. Where is the hereditary information of an organism contained? Human beings have 46 chromosomes in every cell. True or false? Approximately how many genes are there in the human genome? The process of cell division is called mitosis. True or false? Single-celled prokaryotic cells use meiosis to reproduce. True or false? When fertilization occurs, 1/2 of the DNA comes from the and 1/2 from the. Dominant traits are always expressed in the next generation. True or false? How does cancer spread? What is the name of cells that have the ability to develop into different kinds of cells? Why did scientists undertake the Human Genome Project?